This monochromatic model shows a three-dimensional network of neurons firing in a rigid, repeating loop along specific pathways. For decades, this type of anatomical mapping formed the baseline assumption of neuroscience, that neural activity emerges through established mechanisms like synaptic plasticity, recurrent connectivity, and hierarchical organization. These structural mechanisms precisely describe how neural representations are constructed. They show us how individual neurons connect and how those connections form complex, physically wired circuits over time. But relying strictly on fixed wiring introduces a physiological paradox. If cognitive processing is strictly a product of static networks, we cannot explain why identical sensory inputs frequently produce radically different patterns of neural activity. The physical anatomy of the brain remains fixed from minute to minute. In contrast, the physiological and behavioral conditions of the living organism shift continuously in response to internal needs and external pressures. Structural anatomy alone is insufficient to explain behavior. Neural computation is driven by momentary states of excitability acting upon those physical pathways. The solution to this anatomical paradox lies in widespread neuromodulatory systems. These are chemical networks that overlay the entire brain and continuously regulate its function. To understand these systems, we have to clarify what they do not do. Neuromodulators do not transmit detailed information about the external world. They do not encode specific visual, auditory, or cognitive representations. Their true function is entirely regulatory. They dictate the broader physiological conditions under which precise neural computation is allowed to occur. By releasing chemicals broadly across large regions of the brain, these systems alter the overall responsiveness of thousands of neural circuits at once, all without rewriting a single anatomical connection. Neuromodulators do not create representations. They dictate the environmental conditions that force existing representations to compete, allowing specific behaviors and thoughts to emerge based on biological need. Left, neurotransmitters mediate precise single-synapse communication. Right, neuromodulators diffuse volumetrically across broad populations. Observe the graph. The wave modifies resting excitability, pushing the baseline toward the action potential threshold, creating immediate network-wide effects that shift synchronization, increase responsiveness, and alter the excitation-inhibition balance. This mechanism defines a central concept in neurobiology: neural gain. Gain is simply the degree to which a neural population responds to an incoming signal. When neuromodulators increase neural gain, they amplify existing signals and prime existing pathways. They do this independently of whatever novel sensory data is actually entering the system. Shifts in gain determine the intensity with which existing neural representations compete for expression at the cellular level. To see how gain regulation dictates behavior, we can examine four primary modulatory systems. The first is dopamine, a primary regulator of learning, motivation, and adaptive decision-making. Dopamine acts primarily as an error signaling mechanism. It marks the discrepancies between expected outcomes and obtained outcomes, rather than representing the specific details of the reward itself. This error signaling adjusts neural gain to alter the likelihood that particular anatomical pathways will be strengthened, driving behavioral reinforcement through experience. Acetylcholine, by contrast, exercises a widespread influence over attention, sensory processing, and cortical plasticity. Cholinergic activity specifically shifts the balance of cortical processing. It increases the responsiveness of the cortex to external sensory inputs, while simultaneously dampening internal, ongoing neural dynamics. Neither dopamine nor acetylcholine encodes the specific objects you are learning about or observing. They solely adjust the biological readiness of the circuits tasked with processing that specific information. The third major system, noradrenaline, drives arousal, vigilance, and behavioral adaptation under changing environmental conditions. Activation of noradrenergic systems increases gain to prime neural populations for rapid adjustment. It prepares the network to react during situations of high uncertainty or sudden environmental demands. Finally, serotonin operates through a highly diverse set of receptor types and circuits distributed extensively throughout the entire nervous system. Because of this broad distribution, serotonin regulates a wide array of functions, heavily influencing mood regulation, behavioral flexibility, impulse control, and learning. Like dopamine and acetylcholine, noradrenaline and serotonin lack cognitive content. Their purpose is to regulate the conditions under which neural populations interact and adapt. These neuromodulatory systems do not wait in line to perform their tasks. They operate and fire simultaneously across vast, distributed regions of the nervous system. In any given moment, they are simultaneously impacting the sensory cortices, association areas, motor systems, the hippocampus, the basal ganglia, the thalamus, and the prefrontal cortex. This is a volumetric regulation. By blanketing these interconnected regions, the modulators continuously alter the global balance between competing neural populations. A brain's functional state at any given millisecond is the product of its hardwired anatomical organization multiplied by its current neuromodulatory environment. This biological reality creates a phenomenon known as state-dependent processing. Every single moment of cognition occurs within a specific global physiological context. Macro states like wakefulness, sleep, focused attention, acute stress, hunger, or physical fatigue all alter the baseline responsiveness of neural populations. In this abstract network diagram, watch what happens to a single data input under a baseline physiological state, shown here by the blue background. The signal routes cleanly and linearly through a very narrow set of nodes. Now, watch what happens when the background shifts to a red aroused state. Because the neural gain has changed, the exact same data input propagates through entirely different, widely branching pathways across the network. This dictates that core cognitive functions, like synaptic plasticity, the allocation of attention, and behavioral reinforcement, are gated by the broader physiological state of the organism. Identical external stimuli will never guarantee identical neural activity, because neural computation is linked to a biological context that is always in motion. The ultimate principle of neuromodulation is that it is a system of gain rather than representation. Modulators do not encode your memories, your perceptions, or your motor commands. They strictly control the accessibility, the responsiveness, and the capacity for modification of the circuits that do. The nervous system operates as a dynamic computational system whose representations are continuously regulated by biological mechanisms. These architectural blueprints represent a shift from observing organic biology toward modeling the structural frameworks that allow for such flexibility. This biological reality shows how stable anatomical organization and continuously changing physiological states work together, allowing the same neural structures to participate in different patterns of behavior. This chapter establishes how widespread neuromodulatory systems dynamically regulate the responsiveness of distributed neural networks. Tokenization theory later proposes an explanatory framework for how cognition is structured. Moving forward, we will explore whether these established biological mechanisms can be incorporated into the broader framework suggested by tokenization theory.